Hello, my name is Alexa Mogus, and in this presentation, I'm going to discuss the urinary system. Specifically, we are going to focus on water and urea reabsorption at the kidney. So, the urinary system consists of the urine-forming organs, which are the kidneys, and the structures that carry the urine from the kidneys to the outside for elimination from the body. The kidneys lie behind the abdominal cavity and act on the plasma flowing through it to produce urine, conserving materials to be retained in the body, and eliminating unwanted materials into urine. In this slide, we are going to talk about water reabsorption. So water is passively reabsorbed throughout the length of the tubule as water osmotically follows actively reabsorbed sodium. The reabsorption of water in the kidneys is important because it prevents dehydration. Of the water filtered, 65%, or 117 liters per day, is passively reabsorbed into the proximal tubule. Another 15% of the filtered water is passively reabsorbed into the loop of Henle. The 80% of filtered water is reabsorbed in the early parts of the nephron, regardless of the water load in the body. Variable amounts of the remaining 20% are reabsorbed in the distal portions of the tubule. Reabsorption in the distal and collecting tubules is under direct hormonal control, depending on the body's state of hydration. No specific part of the tubule requires energy for reabsorption of water. In this slide here, we are going to talk about aquaporins. Water passes primarily through aquaporins, or water channels, during reabsorption, and they are formed by specific plasma membrane proteins in the tubular cells. There are different types of water channels that are present in various parts of the nephron. The water channels in the proximal tubule are also known as aquaporin 1 channels. They are always open, and they account for the high water permeability of the region. Aquaporin 2 channels in the principal cells in the distal parts of the nephron are regulated by the hormone vasopressin, and they account for the variable water reabsorption in this region. The image on the screen here displays a depiction of an aquaporin, and it shows how water flows passively through intracellular fluid to the extracellular fluid and vice versa. In this slide here, let's talk about the proximal tubule. So on the image here on the slide, the proximal tubule is represented by the color green, as we see here on this nephron loop. So the segment of the nephron that begins from the renal pole of the Bowman's capsule to the beginning of the loop of Henle is the proximal tubule. The proximal tubule is the site where uncontrolled reabsorption and secretion of selected substances occur. The main driving force for water reabsorption in the proximal tubule is a compartment of hypertonicity in the lateral spaces between the tubular cell established by the basolateral pump's active extrusion of sodium. Figure 1419 on the slide here is showing a depiction of water reabsorption in the proximal tubule. So let's recap here. Our main driving force for water reabsorption in the proximal tubule is the compartment of hypertonicity in the lateral spaces. And this is established by the basolateral pump's active extrusion on sodium. So as a result of this pump activity, the concentration of sodium rapidly diminishes in the tubular fluid, and tubular cells while it simultaneously increases in the localized region within the lateral spaces. This osmotic gradient induces the passive net flow of water from the lumen into the lateral spaces, either through the cells or intracellularly through tight junctions which are leaky. The accumulation of fluid in the lateral spaces results in our buildup of our hydrostatic fluid pressure, and this flushes water out of the lateral spaces into the interstitial fluid, where it finally reaches the peritubular capillaries. Water also osmotically follows other preferentially reabsorbed solutes, such as glucose, but the direct influence of sodium reabsorption on passive water reabsorption is quantitatively more important. This return of filtered water to the plasma is enhanced by the fact that plasma colloid osmotic pressure is greater in the peritubular capillaries than anywhere else. The concentration of plasma proteins, which is responsible for hydrostatic pressure, is elevated in the blood entering the peritubular capillaries because of the extensive filtration of water through the glomerular capillary upstream. The plasma proteins left behind in the glomerulus are concentrated into a smaller volume of plasma water, therefore increasing hydrostatic pressure of the unfiltered blood that leaves the glomerulus and enters the peritubular capillaries. This force here tends to pull water into the peritubular capillaries simultaneously with the push of the hydrostatic pressure in the lateral spaces that drives water toward the capillaries. By this means, 65% of the filtered water, or 117 liters per day, is passively reabsorbed by the end of the proximal tubule. And this is seen here on the image, and the blue arrows represent our passive movement of water by osmosis, or our hydrostatic pressure, and the red arrow focuses on the active transport of ions. Next, let's talk about urea reabsorption. So passive reabsorption of urea, in addition to chloride and water, is indirectly linked to active sodium reabsorption. Urea is a waste product from the breakdown of proteins. The osmotically induced reabsorption of water in the proximal tubule, secondary to active sodium reabsorption, produces a concentration gradient for urea that favors passive reabsorption of this waste. Figure 1420 depicts passive reabsorption of urea at the end of the proximal tubule. So at the beginning of our proximal tubule here, in Bauman's capsule, urea is at the same concentration as in the plasma and surrounding interstitial fluid. By the end of the proximal tubule at point B here on the graph, 65% of our original filtrate has been reabsorbed, concentrating the filtered urea in the remaining filtrate, and therefore this establishes a concentration gradient favoring passive reabsorption of urea. So let's backtrack a little bit. So extensive reabsorption of water in the proximal tubule gradually reduces the original 125 milliliters per minute of filtrate until only 44 milliliters per minute of fluid remain in the lumen by the end of the proximal tubule. And this is 65% of the water in the original filtrate, or 81 milliliters per minute, has been reabsorbed. Substances that have been filtered but not reabsorbed become progressively more concentrated in the tubular fluid as water is reabsorbed while they are left behind. Urea is our one such substance in this case. Urea's concentration, as it is filtered at the glomerulus, is identical to its concentration in the plasma entering the peritubular capillaries. Therefore, the quantity of urea present in the 125 milliliter of filtered fluid at the beginning of the proximal tubule, however, is concentrated almost threefold in the 44 milliliter left at the end of the proximal tubule. As a result, urea concentration within the tubular fluid becomes greater than the urea concentration in the adjacent capillaries. Therefore, a concentration gradient is created for urea to passively diffuse from the tubular lumen into the peritubular capillary plasma. Because the walls of the proximal tubules are only somewhat permeable to urea, only about 50% of the filtered urea is passively reabsorbed by this means. In the final slide of this presentation, let's talk about what happens to the unwanted waste products. So in general, unwanted waste products are not reabsorbed. The other filtered waste products besides urea, such as uric acid, creatine, and phenols, typically derived from many foods, are concentrated in the tubular fluid as water leaves the filtrate to enter the plasma. But urea molecules, being the smallest of waste products, are the only wastes passively reabsorbed by the concentrating effect. The other wastes cannot leave the lumen down their concentration gradients to be passively reabsorbed because they cannot permeate the tubular wall. Therefore, these waste products generally remain in the tubules and are excreted in the urine in a highly concentrated form. This excretion of metabolic waste is not subject to physiologic control, but when renal function is normal, the excretory processes proceed at a satisfactory rate. And the average urine volume per day for an adult equals 1.4 liters. And the image here shows what a typical urine concentration consists of, down to each portion of the waste products and the amount that's in the urine.